I want to start by asking you a question. How many of you in the audience know someone with Alzheimer's disease or someone who's caring for someone with Alzheimer's disease? Raise your hand. That's a lot of people. This is a disease that's increasing in prevalence because lifespan is now 80 years old, and at 85, some studies say half of our population at least beginning early symptoms of Alzheimer's disease. So you can imagine what's going to happen. It's going to crush our Medicare system. It's an epidemic that's just going to grow exponentially. Basically, our lifespan has far outpaced our health span, especially our brain health span. You know, in Alzheimer's disease, you have really a thief that just robs yourself of yourself. So all of your loved ones who know who you are and have learned to love you and have basically come to love the reality that you represent in their world, this is all torn away. And the reason for this is that, you know, every day as you have experiences and interactions, you're creating memory maps from when you were a baby. This is the hippocampus. This is the region of the brain that's affected. This is short-term memory part of the brain. Hippocampus means seahorse in Greek. And you have in your brain 100 billion neurons. You have tens of trillions of synapses. By the way, you're born with a quadrillion synapses. And then as the baby grows, those synapses go away because basically living on this planet doesn't require a quadrillion synapses, apparently. So we have about 10 trillion or so synapses, and this is your neural network. And what happens in Alzheimer's disease is as pathology accumulates, this network and everything that's encoded in it, your experiences, your memories with your loved ones, is just basically slowly going to then dissolve away until you're really left with no sense of self or context of self. And this is Dr. Alzheimer, who first described this disease in this patient, August Dieter. And when she was 55 years old, she was brought to what was called the Castle of the Insane in Bavaria. They didn't have very politically correct names for these places back then. And Alzheimer wrote in his journal, 55-year-old woman, memory loss, hallucinations. And the important thing was every night she would cry at the top of her lungs, "I have lost myself. Oh, God, I have lost myself." And that's what this disease is. And the reason for this, and I think this relates back to the beautiful talk we just heard from Divya, is that when we think about the brain and normally functioning, you know, sensory signals are coming in electrochemically, right? So you're getting these electrochemical signals, and that's all they are. But then the brain has to give them meaning. The brain has to put them into context and turn signals into information. So if a baby, you know, sees a red shoe, it means nothing. The baby has to learn that a shoe goes on the foot, who might wear a red shoe. So you have to integrate this information, and then whenever you see something new, you put it into context of what you already know in your brain. Then that information can be recalled later, and you get what's called contextual memory. So if you look at this video, just take a minute. So how many of you can feel the thump a little bit? When you watch this, do you have a contextual relationship in your brain of where you're not just watching it visually, but you can almost feel the ground shake, you can almost hear the thump of it hitting? How many of you experienced that? Okay, so that's because you know what this should sound like. You know what this should feel like. This wouldn't happen for a baby. And if you're a late-stage Alzheimer's patient, you would watch this and you wouldn't be joining together that sensory information. And if you didn't in the audience, don't worry, you don't have Alzheimer's. Not everybody experiences it. It's always a dangerous thing to do this. And so in Alzheimer's, the sensory signals come in and they get actually integrated, but they don't take on meaning. They can't. So if an Alzheimer's patient who's quite late in the disease goes to the fridge and smells rotten food, they're not going to make the connection, oh, rotten food, if I eat that, I'll get sick. They may eat the rotten food. Or they may try to shave with toothpaste because the contextual meaning has been ripped away, starting with the hippocampus, and then it spreads to the rest of the brain. And the reason for that is this pathology that Alzheimer described over 100 years ago, plaques, which are these big bundles of gooey material that are outside of nerve cells, tangles, which are inside, and then neuroinflammation. And neuroinflammation, I'm going to tell you, is the most important part of this disease if we want to stop it. So neuroinflammation is a response to the brain that's evolutionarily conserved since the beginning of humankind, since we first started evolving. And like I told you, our health span is not kept up with our lifespan. Evolution has not kept up with our health span. So we carry the vestiges of an evolution of an immune system in the brain that's at the level of a horseshoe crab or an oyster. In the brain, we don't have antibodies and T cells and B cells. We have primitive peptides that just attack bacteria or viruses when they come in. And we have an immune system with these microglial cells. We heard about them yesterday. Normally, they're trying to clean the brain and housekeep and nurture cells. But when they smell a little bit of cell death, let's say due to a plaque or a tangle, they say, wipe out the grid. It's under attack. They assume the same thing they did 25,000 years ago when you have a 25-year-old who's going to be dead soon, that was lifespan, and if neurons start dying in that 25-year-old's brain in the jungle, it's not Alzheimer's. It's probably because they're having some encephalitis or meningitis. So the brain says, wipe out the grid. Well, that evolutionary program continues today. Those microglial cells didn't get the memo about the Industrial Revolution, the Scientific Revolution, or the Hygienic Revolution. Wipe it out. And that's what kills most of the cells. So I'm going to tell you that plaques and tangles initiate this disease. And you have to hit them early. If you want to help a patient who's suffering right now, you have to stop neuroinflammation. And the good news is we're getting a good handle on how to do that thanks to the genes involved. The first genes I showed there, I was involved with discovering 3 of those. I discovered APP when I was a student, and then presenilins later on, along with others. These are the early onset familial genes. ApoE is the main late onset risk factor gene. Now the problem is, even though all these genes say stop amyloid and you'll stop the disease, for the reasons I just told you, every amyloid drug that's been tried so far has failed. One possible exception, you might have heard about the Biogen news. How many of you heard about the Biogen Alzheimer news? So I'll address that. So the thing is, if you look at this graph, amyloid starts really early on. You see way back there on the left, and you see that first curve coming up. By the time you have dementia, which begins with that first vertical dashed line, plaques have already basically peaked and they're plateauing. And so if you hit plaques there, the question is, can you really do any good? You really want to hit them when they're coming up. It's kind of like cholesterol and heart disease, right? If you have an MI, you have a heart attack, you have congestive heart failure, imagine a cardiologist saying, oh, that's okay, just take some Lipitor and go home. No, you had to do that 10 years ago. We have to hit amyloid also 10 years in advance. Now, Biogen gambled, and they said, what if we hit it right here? Right when dementia first starts, there's a little bit of play in the slope. Might we get the result? And the good news is one of their phase 3 trials did reduce cognitive decline by 23%. A second equally sized phase 3 trial did not. But that might be enough for the FDA to approve it. We'll have to see what happens. That could be the next US approved drug. But ultimately and optimally, you'd want to hit back here. You'd want to have a drug that hits amyloid hitting way back here, the way we stop cholesterol 10 years before you might have a heart attack or heart disease, okay? So this is the elephant in the room, is that we don't diagnose Alzheimer's disease till the brain has degenerated to the point of dysfunction. Imagine if we did that for diabetes. Imagine if we waited till your pancreas started to degenerate and the islet cells are going away, and we say, okay, now we're going to give you a diabetes drug. No, we check for glucose, we check for insulin. Heart disease, same thing. Cancer, we don't wait till you have a 2-inch tumor and try to shrink it, right? But Alzheimer's, we do. Alzheimer's, we wait until the brain degenerates, you're showing signs of dementia, say, okay, now we're going to try to treat you. Well, this is what has to end. So we need to do early prediction. We need to know who's at highest risk, family history. We're trying to build polygenic risk scores using the entire human genome material with whole genome sequencing. And we're not there yet. A lot of direct-to-consumer companies, DNA companies want you to believe we're there, but we're not there yet. There's still very much a work in progress. We also want to do early detection. We need to know when you're in trouble so we can treat you 5 or 10 or 15 years before. And we can do that right now with brain imaging for plaques and tangles, that early initiating pathology. I like to say the plaques are the match. The tangles are the brush fires that spread. You can live with them, as I'll show you. But the neuroinflammation is the forest fire, and that's what takes you out. And the reason why these amyloid trials are failing is you've got a forest fire burning, and you're trying to blow out the match. That's the plaque, or stamp out the brush fires that started it. We can also detect amyloid building up now in cerebrospinal fluid. There's a blood test coming soon from WashU. And then I also work with a company called React Neuro, shown on the right, that is creating a digital device that can do 6 different tests of eye tracking that show whether there's inflammation and trouble in the brain, just like after a concussion, but something that happens more gradually with age with brain health, so we're developing that. So these are just 3 examples of how we might detect problems in the brain early, and especially Alzheimer's pathology. But what can we do about it? Early intervention is what matters the most. So this is the most important slide I'll show you, and I know it looks a little technical. What it's showing is that if you look in the middle at the top 2 panels, you're seeing the amount of plaques and someone who had Alzheimer's disease on the right, and someone who did not have Alzheimer's disease on the left, both of whom had died in their 80s. And then look at the tangles in the next row down. It's about the same. So the question is, how do you get these resilient brains, meaning you have Alzheimer levels of plaques and tangles, but no dementia when you die? And the answer is always the same. This is rare, by the way. These resilient brains, they only come around once in a while. If you notice, under astrocytes and microglia, these are the cells that normally they're helping nurture the brain and clean it, but they can turn on the brain and take it out when they think there's a problem, like they're seeing neurons dying, this evolutionary program I told you about. And you're seeing there that in the Alzheimer patient, you see activated astrocytes, you see activated microglia. In the resilient brain, despite the plaques and tangles, despite the match and the brush fires, no forest fire. And we're doing whole genome sequencing and finding protective gene mutations that are protecting these people. And as a result, in the lowest panel, you see that the resilient person has their neurons intact, the Alzheimer patient's losing their neurons. We now know that neuroinflammation kills 10 to 100 times more neurons than tangles. So when companies are trying to stop plaques, good luck with a patient who's already suffering from dementia. Maybe Biogen got a little lucky with those very early mild cases. And now pharma companies are jumping on the tangles, saying, okay, we'll do the tangles. But you have to look at that diagram I showed earlier. Tangles are also early. It's too late to hit tangles in a patient. We've already seen evidence of this. AbbVie has an antibody, an immunotherapy that stops tangles in another form of dementia called progressive supranuclear palsy. And in that trial, they failed to move symptoms. No surprise. If you want to help a patient, you have to follow the genetics, follow the imaging data, and this neuroinflammation is the target. And this is where genes come in. So on the left side are all the new Alzheimer's genes that we and others have found over the last 10 years or so using whole genome methods. And the two most important ones are the ones that control neuroinflammation. TREM2, CD33. So if you look at this diagram, the blue squiggly thing, that's a microglial cell. Look at the bottom left first. When a microglial cell is doing its job right or well, during deep sleep, it's like a scrubby bubble. Remember those scrubby bubble commercials, if you're old enough? The microglial cells are cleaning amyloid and debris during deep sleep, and you literally squeeze it out of the brain. But when those microglial cells, they're also acting as sentinels, not just housekeepers. They smell cell death here and there due to plaques and tangles. They become now killers. They become warriors. So you look at the upper right. Now they cause inflammation, and they start eating neurons. They wipe out the grid. This is a primitive immune vestige of evolution that we have not yet overcome in our brains. So what we did was we modeled all this, you heard about brain organoids yesterday. This is a little similar. These are actually called 3D neuroglial cell models. So if you look on the left with the spokey thing, in the middle you have neurons and astrocytes and a gel-like matrix mimicking a brain. If you look up here, you can see the neurons are actually forming. They're actually making connections. They're firing at each other. We don't know what they're talking about. It's in the size of a paper punch in a 96-well dish. And then we laser etch in channels where microglial cells in red can come in from the outside, so we can actually track when the microglial cells smell the neurons dying, because we're giving them plaques and tangles with genes, right? So the neurons get the Alzheimer's genes in them. They're making plaques and tangles. Now the neurons rush in, and we can visualize. Now you can see we're making plaques on the upper right. We're making classic tangles. We fully recapitulate this disease. We couldn't do this in mice. This did not work in mice for 20 years, but you can do it in a mini brain-like organoid shown here. And when these the microglial cells, when they rush in, here's what they do. And this is going to loop. See on the right, there's a neuron. Right now, see the axon, that green thread? Look what the microglial cells do. That's neuroinflammation in a dish. That's in a small paper punch-sized mini brain. You're seeing the microglial cells eat that axon for lunch. Now look on the left. See that snapback? That's a synapse that got eaten by a microglial cell, and now the neurite snaps back. So we can actually visualize this, and because we can visualize 96 of these at once in a 96-well dish, we can then rapidly, much more than before, test drugs, natural products, whatever we want to say. What stops the plaques? What stops the tangles? How do we stop neuroinflammation? So if we were using mice, you'd have all those 8,000 mice. It would take 2 years. It would cost ridiculous amounts of money. But instead, you can do this, again, 100 drugs or so at a time in a plate with these mini 3D cell cultures, and you can screen using what's called confocal high-content screening. We've done this. We've come up with a number of approved drugs that do work, approved drugs that stop plaques, some that stop tangles. But like I told you, we're most interested in the ones that stop neuroinflammation. And one of those that we hit is an old asthma drug called Chromelin. And Chromelin, when you took it for asthma, it would stop the mast cells from activating and releasing histamine, causing an asthma attack. Well, Chromelin didn't get into the brain, so we worked with a company called AZ Therapies, and I'm a chair of their SAB, and I am a shareholder in the company for disclosure. And what happens here is that the Chromelin that's made to get into the brain, reformulated, finds those microglial cells, and very similar to what they do to mast cells in asthma, they tell those microglial cells to calm down. So that microglial cell I showed you that's eating the axon and the synapses, you add Chromelin, and it goes back to being a mild-mannered housekeeper eating amyloid instead. It converts them back. So not only do you stop neuroinflammation, but you also promote the clearance of amyloid because you're turning it from a killer back into the housekeeper that sweeps up the amyloid for you. So I think in the future we can think about using this. Another drug I won't talk about today is a gamma secretase modulator. So I think the future will be when I showed you that hit amyloid early, we'll have small little white pills that may be able to do this in the future. Aducanumab from Biogen may open the door to the FDA to be able to bring in less expensive, safer small molecules that can hit amyloid very early, just like we hit cholesterol early to stop heart disease. Now, next question. Should we just wipe out all of the amyloid in the brain, all of the A-beta peptide that makes up the amyloid? And the answer is no. This peptide's conserved for 400 million years. The coelacanth living fossil fish has human A-beta in its brain. This is not an accident. A-beta is not being made accidentally in the brain. It's made for a reason. And what my very brilliant colleague, Rob Moyer, found is that in actuality, A-beta is meant to protect your brain against microbes. That's how it evolved evolutionarily. As soon as a microbe, let's say a bacteria, sneaks in through your compromised blood-brain barrier because you're eating a junk food diet and your microbiome isn't maintaining it. Let's say a virus that you got at 2 years old now activates, like human herpes virus 6 or 7. Why are we not constantly in sepsis? Our brain doesn't have a very good immune system. Why? We take for granted that our brain isn't constantly under a state of meningitis and encephalitis. It's because this primitive system mainly uses A-beta that makes amyloid. And see that yellow spongy, like Spider-Man, You see the microbe, you throw the net around it. That's A-beta will bind to the microbe and then form a plaque around it. In innate immunity, this is called an extracellular trap. In Alzheimer's, it's a plaque. So that's what A-beta is meant to do. It hits viruses, and if you just try to infect mice that are making amyloid with herpes or bacteria, they live a lot longer because the amyloid protects them. That's the upper left graph. And now what we're doing is something called the human brain microbiome map, and we're trying to identify all the microbes that are more abundant in Alzheimer's brain, because if we can understand which viruses, bacteria, or fungus are driving the need for amyloid to protect the host cells by trapping them, we can hit those microbes early on with primary prevention. And that's something we're trying to do. So this is just showing you how we do metagenomic sequencing. And so far, herpes viruses are coming up strong, retroviruses, and we're also finding various bacteria shown in the metagenomic mapping in the middle. The right side is how we look for viruses in the brain. So this is ongoing in our lab right now. So in the future, it will be right patient, right pathology, right drug, right time. Primary prevention might be stopping those infections that drive the amyloid. Secondary prevention will be hitting the plaques and tangles 5, 10, 15 years before symptoms. But treatment will be hit the neuroinflammation, because that's what's killing most of the neurons and synapses. Now we have this understanding, we can go forward. And there are already companies starting to hit neuroinflammation. One of them I just told you about. There are others that are tackling the gene. CD33 is a gene we found in 2008. That's the first neuroinflammation gene. So this is going on. But now I want to end with just talking briefly about what can we do in our lives right now to help maintain brain health and stave off Alzheimer's disease. And for this, in 3 books I wrote with Deepak Chopra, when we go on our little book tours, I had to come up with a way to summarize all of the advice in our books. And one day in the shower, I keep a pad called Aqua Notes, and the pencil says, don't let those good ideas go down the drain. So think about when you're in the shower and the hot water's hitting your head, and you kind of get into a theta EEG, and you kind of meditate, and you get all these ideas, and then you walk out of the shower and towel off and say, what the heck was I thinking about? You know, because it all goes away. So you've got to get the Aquanotes pad, because then you can write it down right away, and it allows you to write with water flowing. So I came up with this acronym called SHIELD, which says these are the 6 things you have to do: sleep, handle stress, interaction with others, exercise, learn new things, and diet. So let me go through. So during sleep, I already told you, when these microglial cells are behaving, they clean all this debris for you. Right after you have REM, or dream sleep, you go into slow-wave deep sleep. That's when you consolidate memories of the day, but also these little microglial cells clean up the amyloid and the debris you made all day. And so it's very important to try to get your 8 hours of sleep. Even if you only get 6 hours of sleep, take naps during the day. Just get those cycles, get those rinse cycles. It's like a rinse cycle. I call it mental floss. Get that crap out of your brain through sleep. It's the easiest thing. But you might have to take naps. Some people can't sleep full 8 hours. I realize that. Handling stress, this includes meditation. Deepak and I published a paper with Alyssa Appel and Eric Schott and others where we found that a meditation trial, and Dean Ornish is coming out in a minute, also found these types of results, that a week of meditation leads to incredibly beneficial changes in gene expression, telomerase activity. And I want to also point out that in regard to the Connectome project you just heard about, if you look at how pathology tracks through the brain over time in Alzheimer's, it follows something called the default mode network. The default mode network is what gives you your ego. Okay? Whenever you're opining, judging, obsessing, you're anxious, you're idle, as opposed to being present, mindful, task-driven, focused, your default mode network is making amyloid. It's making amyloid, it's making tangles. It's almost like an expiration date on your ego. So meditation becomes very important for turning the default mode network off. Loneliness. Loneliness increases risk for Alzheimer's twofold. Not being alone. If you're alone and you like it, that's great. That's probably good. But if you're lonely, you're alone and you don't like it, twofold increased risk. We'd like to be socially engaged people. And exercise. We published a paper in Science last year showing that exercise induces the birth of new neurons in the hippocampus. And the combination of brain-derived neurotrophic factor, or BDNF, plus neurogenesis actually makes Alzheimer's mice better, even though the plaques and the inflammation is still there. You grow new neurons, they thrive, and the combination is to get both neurogenesis and BDNF. So right now we're working with 2 different supplements. True Niagen is nicotinamide riboside to induce neurogenesis, and cat's claw, which is in the Perceptor, is very potent at inducing BDNF. We're testing now these 2 supplements to see whether they can mimic the effects of exercise in protecting against Alzheimer's. Learning new things. Well, obviously, if you make new synapses, right, the more you can lose before you lose it. So remember, in Alzheimer's disease, what correlates most with dementia is loss of synapses. So every time you make new synapses, you learn new things, you're reinforcing old synapses and making new pathways, you're protecting your brain. I tell people when they're going to retire, don't just think about financial reserve, think about your synaptic reserve. Okay? So if you're learning something new right now, I'm protecting you. If I'm putting you to sleep, I'm still protecting you, and it doesn't matter. The last is diet. So the gut microbiome is most important. The Mediterranean diet, which is high in plant fiber, whole grains. Remember that prebiotics are probably more important than probiotics. So this means plant fiber, and there's a very interesting study. Just today, the first Alzheimer's drug in 10 years was approved today in China, not for use here. But it's actually from seaweed. So they're making, this is a drug that's called oligomanate. It comes from seaweed, and it actually acts as a prebiotic in the microbiome to help reduce inflammation in the brain. And this drug literally is just approved as of today, but it suggests that you don't need this. Just eat whole grains, eat high fiber foods. You can even buy prebiotics that come with your probiotic. Nordic Naturals sells a probiotic with a prebiotic in it. So this is all well and good, SHIELD. This is what I call the real mind-body problem. Get up, no. And so you have to be reminded to do these things. So I'll end with just some advice. Just to summarize, sleep more, meditate more, see family and friends more, move more, learn more, eat better, and choose your ancestors wisely. So I'll stop there, and thank you very much for your attention.